institutions already involved, both in Croatia, in UK, even China. We have Wei here from China, who's also part of it. So I think we are making at least some steps forward. So what we are trying to understand in the moment is genetics of protein glycosylation. Because contrary to protein synthesis, where you have a single gene, which will give you maybe 1, maybe 10 proteins with different alternative splicing markers. Now you have 500 genes working together to give you one glycoprotein. But not only this one, but also all the other glycoproteins will be produced by a number of genes which work together. And we know some of them. We know glycosyltransferases, we know glycosidases, but we don't know the others. We don't know the transcription factors, we don't know the ion channels, we don't know the Golgi organisms, all other proteins which are involved. Very indicative study was done nearly 2 years ago in Japan. They were doing a large RNAi library inhibition of neural-specific glycosylation in Drosophila to see which genes are needed to produce normal glycosylation in Drosophila, in neural-specific glycosylation in Drosophila. They identified 109 genes. Only 14 of these genes were the known glycogenes. All other genes were some other genes. So you cannot directly extrapolate because many of those other genes in signal transduction, protein degradation, they will be important for all other types of glycosylation. But I think we can be pretty safe in estimating there's something like 2,000 genes working together to glycosylate proteins. So we're talking about 10% of the genome in a single metabolic pathway, which is by far the most complex metabolic pathway we have. And we know very little about polymorphisms in those genes. And this is actually not surprising, because about common polymorphisms in any disease, we started to learn only a few years ago when GWAS study started. What we know for the glycogenes, we know the CDGs, which either you're dead or you have a leaky mutation. We know the ABO blood groups, but we don't know much about it. But there was this revolution in genetics a few years ago when the GWAS studies were introduced. So people started to look into large population cohorts, look at all genes, and ask the question not, is this gene important? The question was, which genes are important? And this apparently worked. Because when you look into mostly cited papers in Nature, and this is Igor's slide, a few years ago, all those marked with the red arrows are the GWAS papers. So this is something which is being published, which is being published in top-level journals, which is being cited a lot. And fortunately, at the same time, Pauline developed the high-throughput method for glycan analysis, so it was possible to join glycomics and genomics. And we were close enough to jump on the first ship, so we did the first large study of the glycome in humans. It was done in a collaboration between Pauline's and my group. And we published it a few years ago where we showed there is a large variability in human population. And we were actually doing just relatively simple analysis. You cannot analyze all thousands and thousands of different glycan structures. But we did several HPLC runs. And HPLC is good in grouping glycans in similar structures. So we could deduce a lot of information from those few HPLC runs per person. So what we learned, that the glycans are extremely variable between different individuals. But then, in the same individual, they're very stable. So they're more or less predefined. There are slight changes with time, but individual glycome is very stable. Between individuals, there are lots of differences. And there are some individuals which are very different. We call them outliers because they have, this is the normal profile, this person had a huge, this biantenary monocellular structure. This person here had no neutral glycans, or you have different other aberrations. And there are groups of people which have some different glycotypes, which is obviously some kind of a genetic or environmental reason. So, as I said, we can't do it for all possible glycoforms. Even for IgG, this would be very complex. But you can do it in a quantitative way, get some glycotraits, which are specific for individual, and then you can try to put this thing in the GWAS studies. And this is our first attempt. And I think we had only 1,000 people, and we got a very nice peak in here. this was not a fascinating discovery because this was a FUT8 gene, and we found it's regulating fucosylation of this one of the glycans, so it was logical. But this was a proof of principle that it works. So actually, we didn't even publish it, we posted it in Nature Proceedings as a proof of principle that single SNP can have a significant effect on a single glycan. So these things work. We can use glycomics to go to GWAS studies. And then when we analyzed more people, we did the first large GWAS study of the glycome, and we found the first interesting hit, which is HNF1 alpha. And it was linked to a number of glycan structures, mostly related to the absence or presence of antennary fucose. And then with a lot of functional genomics work done in MRC in Edinburgh, it was shown that this gene is actually controlling the number of glycosyl transferases, and that actually this is a master regulator of antennary fucosylation in humans. So actually most of the groups here are the glyco groups, so we know it. So this is mostly for our guests and for our students to learn something new. The idea is to present our current analytical capabilities and some examples how we can do something useful for our clinical colleagues to develop some kind of a collaborative studies. I will deliver something like an introductory lecture to the biology as a field, including some of the examples from our recent work in our lab. So I will start with a typical picture I use to show how things are wrong. So this is something you can find in a textbook. And people believe in this picture, and we know it is wrong. What do we know that it is wrong? We know that these tiny little things, which are shown here, like an addition, do not look like this. Glycans are not tiny little modification on a cell membrane. When you look into the real picture, so when you look into the microscope, electron microscope photography of a membrane, this here white tiny thing, this is a cell membrane. And the big dark layer on top of it, these are the glycans. So glycans are not tiny insignificant things on the surface of a cell. Glycans are covering all the cells, so the picture should look more like this. You have a membrane, you have some proteins, and you have a lot of different glycans on top of it. Glycolipids, glycoproteins, protoglycans, everything there is, it's mostly glycan. So, as a minimal baseline, we should all agree to that the glycans are one of the four principal components of the cell. You have DNA, you have proteins, you have lipids, and you have glycans. And practically anything on the surface of the cell has to deal with glycans. So if another cell is approaching the cell, it will communicate with glycans. If pathogens are approaching the cell, they will bind to glycans. All the hormones, all the signaling molecules, they have to deal with the glycans on the cell membrane before they can actually enter the cell. So the question is, why are glycans being ignored by the majority of the mainstream science? And the answer is not so easy, but I think the simplest answer is because glycans are so complex. They are structurally extremely complex. Normally, proteins, DNA, these are linear structures. You write down the letters and you know the structure. When you think about glycans, you have to think about the branching, the number of branches, the position of branching, the type of linkage, the anomeric configuration of a linkage. There is a huge information capacity in each glycan molecule. And at least 2,000 different glycan blocks can be found on a protein. So compared to 4 nucleotides, 20 amino acids, you have something like 2,000 glycan blocks which are combined in different ways on the glycoproteins, and you get a glycoprotein which is several orders of magnitude more complex than the protein itself. So this is something which is at the moment beyond our comprehension. We cannot analyze it, we cannot understand it in a full detail. Even the single glycoprotein, simple IgG, with one conserved N-glycosylation site in the Fc region, can theoretically be produced in 900 different glycophores. So 30 structures, 2 sites, 900 theoretical glycofunctional and regulatory roles. They shouldn't be ignored. They're important in the majority of diseases, but there's no direct genetic template. So it's a complex genetics. You cannot use simple one-to-one approach. We can use GWAS to identify genes which are in the game, and we should also check whether epigenetic regulation is involved in any specific issue. And at the end, to acknowledge people doing the work. They're mostly sitting here, you know them, so I won't go by detail, but I have a fascinating team which is really doing a great job. And I thank you for your attention.